Dr. Nicole Mifsud, who is a well-known researcher. She's currently the group leader in clinical immunology in the Department of Biochem and Molecular Biology and Biomedicine Discovery at Monash University. Monash received a PhD in transplant immunology in 2004, and she actually was doing a lot of early research here with us and the team and with transplant patients, specimens, etc., way back then, and has been around for some time, is also well known locally and internationally.
Thanks, everyone. It's a great opportunity to talk about the research that we're doing over at Monash University, and obviously that stemmed from some work that I started here many years ago as part of the lung transplant unit. So my research is really focused on T cells and how they play a role in graft rejection following transplantation. And T cells are like sentinels of the immune system. So they're kind of like the little meerkats that pop their heads up and have a look at what's happening around in the tissues. And so you can see in this first sort of panel here is that, the T cells are sort of surveying the landscape. This is what happens where you see your own tissue. You have these molecules called HLA, which Lucy described earlier. And these HLA molecules present these little dots here, which are called peptides. And I'll talk a little bit more about those later. But you can see in that first panel, everything looks normal. There's nothing interesting going on. So there's no interaction between these T cells and that landscape. However, things change. So you can see now in the middle panel, we now have a virus coming in, we have some form of infection, and now you can see that those little peptides have changed colour. Now, this is really interesting to T cells, so they can see them, they know that they're different, and they then are able to interact and form an immune response in order to combat that infection. The situation in transplantation is somewhat more heightened. So you can see here now that we're replacing our own tissue with donor tissue. They have multiple different types of HLA that are being presented when you have lots of mismatching occurring. And again, the peptides, this time, instead of being from a virus, this time they're from their own donor sample cell peptides. So again, the T cells are interested in this landscape, they bind, they're activated, and they can produce very strong immune responses that can destroy the graft. So in the laboratory, we've been working on looking at these T cells for a number of years now. And what we've been able to do is really identify which are the T cells that are able to respond to these HLA mismatches. And we do this using a particular assay called the mixed lymphocyte reaction. It's basically an assay where you take some recipient cells, you mix them with the donor cells, and you wait to look for the responses. And we can generate different types of T cells compared to the different mismatches that we see between the donors and the recipients. We can assess this using a standard technique, as you've heard before, using called flow cytometry. So this is very well characterised in our laboratory. At the same time, we get information not only about the T cell, but about the HLA molecule that they're responding to. So you can see here that we can quantitate which of the responses are much more likely to be, triggering that immune response. What we can also do is look at this T cell receptor. So a T cell receptor is the part of the T cell that actually interacts with both the HLA and the peptide. And we can do this at a very high sensitivity level. So we can take single cells now, we can look at the sequences of those T cells, and we can also look at their gene expressions. We get lots of information from that end. And we can also look at the relatedness, so how many of those alloreactive cells are able to trigger that immune response. What we haven't been able to do, and what has really been the bottleneck in this whole system, is identifying those dots. So what are the peptides? How are they triggering the T cell responses? Are we looking at one peptide? Are we looking at a whole handful? And so we use a technique called mass spectrometry. And basically what that means is that it breaks the peptide up into different fragments, and we're able to bring those back together and assign a particular sequence to those. So this is what your cell looks like, maybe not so simplistically, but in the middle of the cell here we have a system where we can take some of our own proteins, or those that from the donor cells, they've got their own proteins as well, and then we shuttle them through a barrel-like structure called the proteasome. And in the proteasome, what happens is it trims these proteins, and at the end, what liberates out from the proteasome are these peptides, and we get lots of different types of peptides. These peptides are then loaded onto these HLA complexes, and then they're shuttled to the cell surface. And this is where the T cells recognise them. But what are these peptides? So we know that they're small protein fragments, but they can come in a whole heap of different flavours. So, we can have linear peptides, and each one of those circles represents a single amino acid. So when you put a whole heap of amino acids together, you can get a peptide being formed, and you can see that they range in different kinds of lengths. These peptides can also be modified, and this may happen from a chemical reaction that may occur. And so you can see that these have got either phosphate residues, cysteine residues, or acetylation residues that are also compacted on top of these peptides. Peptides can also take the form of two proteins that have come together and been stitched together. So you will have one side of the segment coming from one protein and the other side of the segment coming from a different protein. So it's really quite a complicated system. And all of these proteins together come together to form what we call the immunopeptidome. And for those that know scientists know that we like to complicate matters, we like to have these very long names. So we're trying to investigate the immunopeptidome of donor tissues to see if we can identify what peptides are actually triggering responses. So how many peptides are actually on top of it or being expressed on the surface of the cell? So it was estimated back in the early 90s that up to 10,000 different type of peptides could be represented on a cell. And we know that with higher sensitivity instrumentation and technology that we have today, it's much more than that. And so we now have the capacity to evaluate that at a much greater level. So we're very lucky in that we are able to access some donor material from the Australian Donation and Transplantation Biobank. And this is where we are able to get different types of tissues from the donors that have been research consented. And this is really important because we have two different categories, obviously, of thank you. And we have two different categories of donors. So we have, donation after brain death, and we have donation after circulatory death. And even those end-of-life events can actually change the immunopeptidome. So some of those peptides can actually change, and even the landscape on top of the cell will change, and so T cells will see something different. And so we really need to be able to evaluate that. We get really nice information from the biobank regarding the demographics of these donors, and we also get some very useful clinical information about what their viral history is, some of their comorbidities, and even some of their lifestyle choices. Because again, things like smoking can also change the immunopeptidome. So how do we go about this? So we basically are able to get these donor tissues. We take very small pieces, just a very small chunk, the size of a pea or a little bit more. We grind them up into a powder, and then we use very standard biochemical techniques to able to extract out the HLA molecules using antibodies and as well as the peptides, and we dissociate these complexes. We can then just take those peptide fractions from each of those organs, and then we can run them through this mass spectrometer, and we can assign a peptide sequence to each one of those. And as you can imagine, we generate a lot of information. A lot of data is coming out of this. So we're not just getting information about the HLA molecules, but all the different type of flavours of peptides that are there. And we also get some information about gene expression as well. So we really need to house that information somewhere. So we've now developed a program called Atlantis, where we can have all this experimental research data, and we can marry that with the donor data, and we're also looking to extend that out to the recipient data very soon. So Chen here is a bioinformatician in our laboratory that we've been generating this program with, and Sanjay is a master's student in data science, and he's absolutely exceptional, and in a matter of 3 months he's been able to work up the prototype. So I'm just going to show you a little bit of information about what we've been doing in terms of the preliminary data. So Ifra is a master's student who's just finished up in our lab, and Murad, who's here today, is also a postdoc in the lab, but also he's a medically trained. So, we've been able to very carefully look at the anatomical features of all of these tissues and make sure that we're very controlled in the way that we're analysing the data. So here we've got data information from 4 different donors. We know they come from different types of categories of in either DVD or DCD. And this is a lot of information about HLA, probably not so much that you need to understand. But what you can see is that, 4 of these individuals co-express the same HLA. And so we're really interested in looking at that in the first instance, but we're also gathering all the information that you can see as well. In terms of the tissues, we've got a few different tissues from these different donors, and that's really good when we want to do comparisons between them in looking at their peptide repertoires. So this is really just a very, it's a busy data slide, but I'll walk you through it. But so we use a particular antibody to try and attract the HLA-A2 molecules. And you can see across the 4 different tissues here is that most of the peptides are 9 amino acids in length. And that's what we would expect. The good thing about this is that we can take those 9 amino acid peptides and we can assign whether they're really strongly bound to HLA molecules or weakly bound. So anything below this dotted line is a strong binder, anything above this dotted line is a weak binder. And we anticipate that the strong binders are the ones that are likely to be able to trigger those T cells that can then go on to form destructive responses against the graft. And then the sequences that we see down here is just to confirm that they are actually, coming directly from the HLA-A2 molecules. So what are the questions that we can ask with all of this data? So we can ask things like, for any particular donor, when we want to look at all the different types of tissues, how many of those are unique to that tissue and how many are commonly expressed across all of the tissues? And so you can see here that, there is a large amount that are unique, but there's also a fair amount that are also common. We can also ask questions about different types of tissues across a multitude of donors. So we have here 3 different donors. And if we're looking just at heart-specific tissues, again, we have unique kind of peptides, but we have a common set that's also available right across all of those 3 donors. And then we can ask the biggest question, which is if we take all of our donors, all of our tissues, how many common peptides are there? And so we can whittle that number down to something that's a little bit more amenable to more scientific interrogation. So we've got now about 74 different peptides that we're hoping to now look in the laboratory to see if they can actually trigger T cell responses. Because this is important because it's not just against the lung or the heart, but we can look across a range of different tissues, which will give us much more information when we're looking at trying to design immunotherapeutics to target these T cells. So why are we doing it? We want to really bring down an approach where we've got the current strategy where we're using anti-rejection drugs or immunosuppressants that are really dampening down the whole immune response. And as Greg mentioned, what this does is it is a fine balance between, trying to maintain a normal immune system and then also trying to stave off any rejection episodes. But of course, that leaves our patients vulnerable to infections and cancers. What we want to do is we want to try and tailor it a little bit more so we can design immunotherapeutics so that we can target these specific T cells that are causing the damage, leaving all those other immune cells readily available for normal immune protection. And again, that is obviously our ultimate goal. So there's lots of people to thank. Obviously, I've tried to thank those along the way in terms of the slides, but of course we've got lots of platform facilities that we've been utilising. Of course, the Alfred Hospital, the funding agencies that we've been able to build our small seed funding on over the years. And that's where we think Lungitude is really important because we can take very small sets of preliminary data that we're generating now and we can then go to our major government sources and try and access more funding. So that's all I have to say, and I'm happy to answer any other questions.